Hello dear students, welcome to Practically. We bring learning alive, and in today's class we are going to discuss the solution for yesterday's board exam of biology for class 12. I hope your exam went well, and let's discuss the solutions for the questions. I know, we say that you should not immediately discuss the solutions, but this board exam is not just for your boards, but the concepts which were asked in the exam, they will also be useful for you in NEET and other various competitive exams for biology. So let's discuss the solutions. Beginning with the first question. From section A, the question was, give your interpretation of the message given by ecologist Paul Ehrlich through his analogy of river popper hypothesis. A very direct question from NCERT. So we all have studied river popper hypothesis. So we have to just explain it. It was for 2 marks. So any 2 important points will fetch you full marks. So we need to first mention the interpretation of message given by Paul Ehrlich. So first of all, we need to explain that what was his hypothesis for, or what was it about. Okay. So it was to show the effect of biodiversity loss. So this is the keyword over here, effect of biodiversity loss on ecosystem, with an example of airplane which has thousands of rivets like screws kind of structure. So popping of rivets, which is equivalent to what? Species becoming extinct. Okay. So this is what this entire part will explain, the rivet popper hypothesis, what it is, that we need to tell in short, that if some rivets pop up, it might not affect the flight safety, but it will become dangerously weak over a period of time. If more species get extinct, if more rivets get popped up, it can become more dangerous. And removal of rivet is critical part of wing, so it can affect the ecosystem. These things we have to explain in this answer, and you will get complete 2 marks for it. Okay, moving on to the next question. State the impact of constant mechanical agitation and pumping of air in aeration tank on the sewage during biological treatment. From the Microbes in Human Welfare chapter. So again, it was for 2 marks. So in this particularly, we have to give the importance of mechanical agitation and pumping of air. So we know that biological treatment is a secondary treatment, and in that air, the effluent is passed through large aeration tanks with constant mechanism supply. What does it do? It helps useful aerobic microbes to grow rapidly, because aerobic microbes will need oxygen. So if we pump in sufficient air, they will grow rapidly and form flocks, and growing microbes consume organic matter and thereby reduce the BOD of effluent. BOD, the biological oxygen demand. So it is actually going to help for the treatment of the water, for it to become more clearer, more pollution free. So we have to just mention these 2 points and you will get 2 marks. Next question. Cattle excreta is important source for producing a domestic fuel. Name the fuel and write its main components. So this question has 2 parts in it, A and B. B says, write the biological process that is responsible for production of this fuel. Okay, again simple question. So we know that biogas is produced by breakdown of organic matter such as food and cattle waste, excreta. And it can be used in various ways, as we know, as a vehicle fuel it can be used. It can be also used for heating and electricity generation. And what all things are contained, this is like additional, you need not mention like all details, but yeah, the important component is methane, which is 50 to 75%. Apart from that, we have carbon dioxide 25 to 50% and small amount of nitrogen. Okay, trace amounts of H2S, ammonia, hydrogen. These things we need to mention, and various volatile organic components etc. are present in that. So that is the composition of biogas. This was answer for A. Now B says that write the biological process that is responsible for production of this fuel. The biological process responsible for fuel is called anaerobic digestion. The digestion of these substances when happens in presence of anaerobic microbes, those who do not depend on oxygen, that is the main process of formation of biogas. And it occurs naturally also in digestive system of various ruminants, organism, and also in various ponds, etc. Next question, it's a data interpretation based question. For looking at it might look little tough, but actually it's easy, we can easily solve it. Let's see how to do it. Is the histogram, which like basically tells the graphical representation of a certain phenomena, representing data for annual shark harvest in Great Barrier Reef or Coral Reef located on east coast of Queensland, Australia. So we have to study this histogram and answer the following questions. So this shows catch in tons on y-axis and it shows the year. Okay. So write your interpretation of the following given data. So what according to this data, what do we find? Just analyze the histogram and what do we find? That the rate of annual shark harvest in Great Barrier has increased exponentially with a minor dip in the year 2002. So right from 1992 if we see, there is a increase, right? With a minor dip in 2002, there was slight dip, but otherwise it has been continuously increasing. That is what the interpretation of this data is. Then write the impact of biodiversity of the area that you can interpret on basis of given data. What will be the impact on biodiversity of that area? It can result in extinction of shark in this area, creating imbalance in biodiversity. Yeah, so if there is extinction, then it can cause imbalance in biodiversity. That is the answer for this question. Moving on further. Stability of community depends upon its species richness. How did David Tillman show this experimentally? Well, he showed this by using outdoor plots. Okay, outdoor plots he did, and each plot had different number of species, and Tillman found that the plots with more species showed less year-to-year variation in total biomass. And he also showed that the experiments that increased diversity contributed to higher productivity. So this is the keyword over here which we need to mention, and it will easily get 2 marks. Okay. Moving on to the next question. What is the importance of female Anaphylus mosquito in the life cycle of malarial parasite Plasmodium? Again, a direct question. Just we have to explain the importance of female Anaphylus mosquito. It is a part of the entire cycle which goes on. So yeah, so we know that a female mosquito is the one which is going to take the blood meals for egg production, and these blood meals are the link between human and the mosquito host in the parasite life cycle. So that's why the female Anaphylus mosquito plays an important role in the life cycle of this parasite. That is the connecting link between humans and the parasite. And yeah, another thing what happens in mosquito's body is the sexual reproduction of Plasmodium takes place inside it, while man is the intermediate host. Yeah, and also gametocytes are in human blood, but sexual reproduction takes place inside female mosquito. So that is the significance of female Anaphylus mosquito in the life cycle of malarial parasite Plasmodium. Okay. Next question of 2 marks. Mention the parts of human body that get affected by pneumonia and common cold infection. Write the causative agents of these 2 diseases. Again, very simple and direct question. Just have to write the parts getting affected and the causative agent. Well, we know that lungs are the one which are going to get affected. Air sacs to be particular, pneumonia affects that. It gets filled with fluid or pus, causing cough with phlegm or pus, and then there are other symptoms. Symptoms are not needed to be right because they have asked the parts of human body getting affected. And the causative agent is Streptococcus pneumoniae. Okay, then common cold, the parts which get affected is nose and throat mainly, and it is caused by various viruses, but rhinovirus is the most common cause. So just half mark for which part, half mark for the causative agent, again half mark for the organ of common cold, and half mark for the causative agent of common cold. So like that you can easily score 2 marks. Okay, moving ahead to section B. Now we have 3 marks question beginning from here. First question, read the amount of natural large forest area that underwent many changes from the year 2002 2014 and answer the question that follow. Okay, in 2002 there was a large area of natural forest habitat in country. In 2007 a canal was built midway through this natural area. This was required to meet irrigation demand of nearby areas. In 2014 ecologists suggested to create green corridors connecting two fragmented parts of the initial large natural forest habitat. Explain the reason behind such suggestion by ecologists. So it is again a question in which you have to interpret whatever data is given and explain in your own words. Such kind of questions are a bit tricky, but if you know the concept, you can explain it in your own words. So what all we need to write, that green corridors are attempt to mitigate negative effects of the built environment of cities and towns, because you know whenever there is any thing which might disturb the habitat of organism, it does affect that and affect the biodiversity. So these green corridors are like attempt to minimize the negative effects which cause. They also act as conservation for rapidly extreme intervention and development. Okay, they act as conservation, and also it enables dispersal movement of animals within city areas. So these are the 3 points which you have to mention, 1 mark each, and you will score 3 marks for this question. Okay, understood? If any doubt or query, please feel free to type in the chat box or comment, and we will be there to help you out with it. Moving ahead, enumerate the main sources of biofertilizers, giving 1 example of each. Yeah, so now since this is a 3 marks question, it is implied that at least 3 such sources of biofertilizers we mention along with the example. Okay, so first one, so biofertilizer is a type of fertilizer containing living microorganisms that enrich. So this is just a short introduction about biofertilizer which is given. Main sources of biofertilizer are bacteria, fungi and cyanobacteria. So already we have mentioned 3, and then we are going to give example of 1 by 1. So nitrogen fixing bacteria are of 2 types, symbiotic, which is rhizobium, and loose association, that is azospirillum, and there are also free living which are Clostridium. Okay, so these are the examples already covered. Then Anabaena nostoc, cyanobacteria which I mentioned, they are important cyanobacteria. Examples Anabaena nostoc, and they also form an association with certain plants and are responsible for nitrogen fixation. That's why they act as a biofertilizer, because plants require NPK, these 3 elements in very important, so that if anything contains these 3 elements, that acts as fertilizer. And fungi example is mycorrhiza, which is also a kind of form symbiotic association. They help in absorbing phosphorus and also increase surface area for absorption of water and other nutrients. Okay, so these 3 things we have to mention, and you will easily score 3 marks for this question. Moving ahead, explain, giving reasons, the action plan followed by organic farmers that support their key belief, biodiversity furthers health of croplands. If you will observe, this time CBSE, what they have done is, the paper is like kind of more of a discussion based paper they have given. It's like you might not find lot of direct question. They are more of a there is some data or some story going on. So that shows that the CBSE is moving more towards application based question, and that's why for the upcoming NEET exam also we should be alert and prepared of attempting such application based question rather than memory based question or something to memorize. So I know the NEET is coming closer. You have already studied the topics. Try to understand the concept and solve more application based concepts and questions, and we are there to help you out for that. So please stay tuned with us on Practical 11th and 12th channel. We'll be also helping you out for your NEET exam preparation. So stay tuned with us. Okay, moving ahead with this question. So yes, organic farmer indeed believes that biodiversity furthers health. It depends on composting and using natural ingredients in conducting this cultivation. And there is no use of fertilizers or chemicals. That is what the feature of organic farming, main feature of organic farming is. And crop cultivation is done in all natural environment. Then the organic farmer also uses biodiversity to counter the problems of cultivation. And by this, it also helps in overall health improvement. And since this is completely chemical free, no chemical is used, only natural produced is utilized, and that's why it increases health of all, even the plant, the land, and people who are going to consume the food. It's healthy for all of them. So that's why this statement that biodiversity furthers health of crop plants is apt in organic farming, and this way you can explain that. Okay, let's move on to next question. State the role of a selectable marker in rDNA technology. Question from biotechnology. 3 parts it has, one by one mark each we have to answer. Let's see the first one. State the role of selectable marker in rDNA technology. Well, we know that selectable markers are important to differentiate or to find out, identify and eliminate non-transformants and selectively permitting the growth of transformants, that is the host cells containing recombinant DNA. That is the function of selectable markers. Simple, direct. Name one such selectable marker which is considered to be useful for E. coli. One such selectable marker, there are many actually. Antibiotic resistant genes like ampicillin, tetracycline, chloramphenicol, kanamycin, etc. Then give one reason why it is considered to be a useful marker. Because antibiotic resistant genes used as selectable marker in E. coli, as it does not contain antibiotic resistant gene naturally. Okay, so that's why it is considered as a useful marker. So again, one mark each, and you will score 3 marks easily. Okay, moving on to the next question. This was a OR option question for this. What are plasmids? How are they different from cloning vectors? Give one example each for a viral and a bacterial cloning vector. Okay, so plasmid, the standard definition, small circular double stranded DNA which is extra chromosomal word is also required, distinct from a cell clone. That is what extra chromosomal is. Plasmids naturally exist in bacterial cells and they also occur in some eukaryotes. Then plasmid are extra chromosomal, it is mentioned here. And cloning vectors are artificially introduced into DNA molecules into the cells, right? And example of viral is bacteriophage, is a viral cloning vector. Bacteriophage, which the virus which infects bacteria, that is bacteriophage. And cosmid is an example of bacterial cloning vector. Okay. Next. Name any 3 techniques used for introducing an alien DNA into a host cell and mention how. So as the question says 3 techniques, so simple, one for each. And here are the 3 techniques. First is indirect gene transfer. In this, what happens is, it is basically a gene transfer is indirect because the DNA is inserted into host cell by use of a vector. So vector is like a vehicle. So that's why it is indirect way of transfer, gene transfer. And that happens through, it can happen through plant tumors or viruses. Okay, it can be either, so we know the Agrobacterium tumefaciens is used, very commonly used for plants, and viruses are used to carry the foreign gene and then infect it with its own genetic material. Then second method is direct gene transfer. It has further subtypes in it, like heat shock. As you know, we first keep the cells on the ice, 0 degrees Celsius, then raise the temperature at 42 and then again keep them on ice. So heat shock increase the size of pores so that the DNA enters in it. Electroporation, by passing high voltage pulse, we make sure that the cell pores, the membranes in the pores in the cell membrane increases so that the DNA enters in. Gene gun, mostly used in case of plant cells where the tungsten or gold particles are covered with the DNA and they are shooted into the cell directly. And micro injection, mainly used for animal cell, where we directly inject the DNA into the cell. So these are the different methods of direct gene transfer. Moving on to the next question. Biodiversification of a life started to occur almost 3 billion years ago. Since then, new species have been evolving and then disappearing from Earth. How many episodes of mass extinction of species have already taken place, and which one is in progress in the current era? How is current episode in progress different from previous episode and why? Explain. Okay, so this was again a little bit no, I'll not say tricky question, but this was a little uncommon question. Such type of question has not been much asked before. So yeah, we can again predict such more such questions coming up, because CBSE won't keep on asking previous year question always, but we need to be prepared for all type of question. So here is the answer. There have been total 5 mass extinctions till now in Earth's history, and the worst one 250 million years ago. 96% of marine species and 70% of land species died off, and obviously it took millions of years to recover. And currently we are at the 6th episode of mass extinction. Now the answer, why? Like, how is current episode different from the previous episode and why? So the previous episodes were caused by natural phenomena. Obviously, like for example, the meteors fell and then there was, like because of falling of the meteor, the Earth's climate and other situation changed, and a lot of species got extinct. So, or there was volcanic eruption. So these are all natural phenomena. But 6th mass extinction is driven by human activity. So we ourselves are responsible for taking us towards mass extinction, which is really an alarming thing. So because of unsustainable use of land, water, etc. And then that's what, like, there's a lot of things, but you can explain in your own words that how human activities have been affecting our planet and us, and slowly it will cause depletion of natural resources and can lead to 6th mass extinction. That's what you have to explain in your own words, okay? And then you will score 3 marks easily. Moving on to the next question. Again, this is a data interpretation based question. Section C, case based, the last part of the question paper. To save the crop plant from the attack of various insect pests, the biotechnologists have developed many pest resistant plants. One such example is Bt cotton. In this plant, cry genes were introduced, which produces cry proteins in a plant that has toxic effect on pests, thus saves the corn plant from attack of corn borer. An experimental field study was conducted by scientists to see efficacy of Bt corn plant against the attack of corn borers. 3 different species of corn borers, namely A, B and C, were collected and were independently fed on non-Bt cotton, that is Bacillus thuringiens, corn plants, and Bt cotton plants separately for the same period. The extent of damage caused to the leaf area of the plant was observed and noted down. With the help of observation and data collected, the following bar graph was plotted. So again, there is another graph. Y-axis shows the percentage of leaf area damaged, and x-axis shows the type of plant. These are the non-Bt plant crop, and this is the Bt crop. Okay, A, B and C. So based on this, we have to answer the question. Here's the first question. Identify the species of corn borer that was most successfully controlled by Bt corn plant. Give appropriate reasons for the interference. So yes, So which, it's very direct question. From this, what can we understand, which was most effective? We have to see the graph, and wherever there is maximum difference, okay, in that is the most effective. So you can clearly see here, this one, that is A, it was almost 80 in non-BT crop, and once BT was introduced, it almost fell down to 10. So the maximum difference is in A, isn't it? So that's what the answer for this question is. The species of corn that was most successfully controlled is A. And give appropriate reason of interference, the same thing you have to tell, that it was 80 and then it went down to 10, which is comparatively higher difference than the other, B and C. Okay, simple. Second, identify the species of corn borers which shows least impact of toxin produced by BT genes. So it is like opposite to the previous question. In previous we saw the maximum difference. In the next question we have to see the minimum difference. That is, which is having the minimum difference? It is the B. B, before it was 20, after using BT cotton it is little less than 20. So B is having the least impact of toxin, that is B. Third question, what would be your advice as a scientist to the farmers for growing this particular BT cotton variety in the area which is infested by species B of corn borers? So to the farmers growing this particular BT corn variety in the area which is infested by species B of the corn borers, so what advice can we give to them is like, since the B is least impacted, so we need to use other measures also to prevent the corn borers, because by BT, using BT technology or thing, it is not able to control much, so we need to use other techniques as well. Name one BT gene that encodes protein in corn plants to control corn borers. Well, as we all know, it is the CRY1AB gene. This is a gene which encodes protein to control them from corn borers. Okay, so this was question from case study. There was one more question as or option for the case study. That is, a gene was identified in a fungus by a research worker in a lab which was considered to be of great importance in the field of agriculture. As a student of biotechnology, write the steps you would suggest to isolate this gene of interest from the fungus and amplify this gene for further experimentation and research. So what are the steps? Simple. We have to write the steps of the recombinant DNA technology. So first is isolation of DNA. Since it is a nucleic acid enclosed within nucleus, isolation of DNA is not an easy task, and for that we need, it is an enzymatically controlled process for which we need to use certain enzymes like cellulase for plant cells, lysozyme for bacterial cells, and chitinase for fungal cells, which will break the cell wall, and then we can isolate the pure DNA from the cells. Then once the DNA is isolated, you need to cut the desired gene of interest, that is fragmentation. Cutting will be done by restriction endonuclease, which cut at specific sites which we want, and then they can be used in the recombinant DNA technology to detect the location of that particular gene, and then they will identify the palindrome sequence and then snip the DNA at a particular part, and then the desired gene and the vectors are snipped by the same restriction enzymes, and then we join them using ligases, and then we get the further process. Next process is amplification of gene of interest. Since the gene of interest which we obtain, it will be very less in amount, but we need to increase it more in amount. That is best done by PCR, polymerase chain reaction, to amplify the gene of interest. And we know the process of PCR. It has 3 steps in it: denaturation, annealing, and extension. So that will be carried out. So yeah, that was all for this question paper 2022 biology CBSE. The paper was indeed very interesting and different compared to previous years. It was more on an easier side, but it was little bit lengthy because the questions were long and more interpretation based. And I hope you all have solved well. And if not, don't worry, you still have more opportunity for the competitive exams which are coming up. So do prepare well for that. And thank you for watching. See you all next time. Stay tuned with us because we are going to come up with more such amazing concepts, topics, question solving for your competitive exams as well. Okay, so thank you for watching. Do subscribe to Practically.